Hey everyone, thanks for jumping on with me. Just wanted to talk a little bit about chromosomes. So, hopefully, at this point in the year, we know that our chromosomes are found inside of the nucleus, and the nucleus is the control center and holds all of our genetic material. So, when we're talking about chromosomes, one of the things that you need to know is how many chromosomes a human has. So, at this point in the year, you should know the human magic number is going to be 46. So, if we have 46 chromosomes, that's going to be 23 pairs. When we're talking about our pairs, we're getting one pair from mom and the other from dad. So, you're getting half of your DNA from sperm and the other half of your DNA from the egg. Chromosomes contain DNA. So this all comes back down to our dark green cars never crash. So DNA makes up your genes make up your chromosomes. The chromosomes are found in the nucleus, and then the nucleus is the control center of our cell. Now there are two types of chromosomes. So you're going to have your autosomes, or you're going to have your sex chromosomes. Now, autosomes are going to represent numbers 1 through 22. The 23rd pair is typically referred to as the sex chromosomes, and that's going to determine gender. So remember, XX gives a female and XY gives a male. So because males have an X and a Y chromosome, the male really does determine the gender of the baby, because half the sperm are going to have X's to give and half the sperm are going to have Y's to give. For females, all we have are X's. Now, autosomes, again, are going to be your 22 pairs. It's number 1 through 22. And those are going to determine everything that has to be going on in you, from your hair color to your eye color to whether or not you can curl your tongue, if you have attached earlobes or non-attached earlobes. Everything that's going on with you is going to be part of those autosomes. Now, the sex chromosomes, again, I just said this before, if you're a male, it's XY. For females, it's XX. Now, the X chromosomes are typically larger in size than the Y chromosomes. The Y chromosomes don't look like a Y. They just are named because they're a little bit smaller. So it's nothing with the way that they actually look, even though we know that a duplicated chromatid looks like the shape of an X, but the Y chromosome doesn't look like a Y. Now, an amniocentesis is really, really important when we're going to be taking a look at the chromosomes. Now, in order to look at chromosomes, you have to do what's called a karyotype. Now, a karyotype typically is going to look at the metaphase stage of a chromosome. When our chromosomes are in metaphase, they are duplicated. So they're going to have two sister chromatids that are attached at the centromere. Now, with this amniocentesis, it's a large needle. And if you look at the picture that's on our page right now, the amniotic fluid is taken from the needle, and then it's actually going to be analyzed because the baby's chromosomes are found in that amniotic fluid. Now, the amniotic fluid does help to protect the baby. It allows some cushion. And when you're doing your karyotype, we're going to be looking at the homologous pairs, and we're going to line them up 1 through 23. So you try to match them up, number 1 with number 1, 2 with number 2, and so on and so forth. And again, when we're looking at our homologous chromosomes, you're looking at the same size, the same shape, and the same genetic material. So if you were to have the ability to curl your tongue on chromosome 3, it could be a yes or a no. On the other chromosome 3 would also have in the same spot the ability to curl your tongue. They don't have to both say yes or no, but it's the same trait. So again, with our amniotic fluid is going to be taken through the amniocentesis. Here's a picture of the baby. You're going to have to use a sonogram because you don't want to pierce the baby. And then our karyotypes. Now, the karyotypes, there's a couple of questions that they're able to ask you, and I'll get to that in just a minute. But your karyotypes are going to be the lined up 46 chromosomes. That's 23 pairs. You can see in the picture that's shown here, they're going all the autosomes first, 1 to 22, and then our last spot has our X and our Y. Now, this is going to be able to help us determine some chromosomal disorders. Now, in terms of our purposes, what we need to know is we have to be able to look at spot 21. So if we're looking at spot 21, it's going to let us know if there is trisomy 21, which is also known as Down syndrome. So again, our chromosomes are going to be numbered 1 to 21. Number 1 is going to typically be the largest chromosome, and little by little, they're going to move until they get smaller. Again, take a look at X. X is going to be larger than chromosome 22. And then our Y is always going to be last if it happens to be there. For our purposes today, right now, this one has 2 X's and no Y's, so we know that this is going to be a female. So here are the questions that they're able to ask you. They're able to show you a picture of your karyotype and simply say, what is this? So if we're looking at our karyotype, it's a karyotype. They can also ask you to look at that last spot and determine the sex of the organism. So if you see 2 X's, it's going to be a female. If you see an X and a Y, it's going to be a male. They can ask you to diagnose whether it's going to be a normal individual or an individual that has Down syndrome. So for that, you would have to look at our 21st spot. And then they can also ask you about the procedure. So if they ask, how do we get a karyotype? You need to know about the amniocentesis. So hopefully this helps you guys. And if you have any other questions, please reach out to me via Remind or Google Classroom. I am also going to post another video about how to analyze the karyotypes in a little bit more detail with some practice, as well as a practice worksheet. All right. Thanks, guys.